Good afternoon, everyone.
Hello.
Good afternoon.
Afternoon.
Hello.
Welcome back. It's nice to see you all. Okay, well, good afternoon and welcome back, everyone. So, as you know, we will have our first lecture exam of the semester this coming Thursday. Okay, so just so there's no surprises there, right? So I'll be sending out a class email with instructions that I'm going to tell you right now, but basic process is on Thursday. Hold on, let me get some of your classmates in here. Okay, so on Thursday, you'll log into classmarker.com at 12:30, the start of our normal lecture period, and you'll have 1 hour and 15 minutes to take your first lecture exam. Okay, so Thursday, log into classmarker.com at 12:30, take your exam, and then after you're done with that, we do have lab Thursday afternoon, so please join me on Zoom at 2 o'clock for our normal lab time. Okay, Samantha, I see your comment to me and I understand. Okay, I hope you will feel better. Okay. Okay, hold on a sec. I need to take a look at some chat messages to me here. Okay, Tiffany, let's talk a little bit after class today. Okay, it won't take long. Okay. All right. Okay, so before I get started lecturing in chapter 8, if you do have some big content kind of questions you want help with for the exam coming up, be happy to do that during office hours this afternoon. But are there just any quick burning questions you really just need to ask? Okay, reach out to me if you're having a problem. Come see me. I have office hours today, tomorrow, and even Thursday before the exam. I have office hours, so pop in if you have a need to do so. Okay. All right. So you will not need a calculator for the exam on Thursday. Okay. So those that have been feeling some stress about the math and lab and things like that, you do not need a calculator for the exam on Thursday. However, next Tuesday, a week from today, I will be giving a rather mathematical kind of lecture on a topic called unit conversion. So I would ask you at that time to have your scientific calculators available. At the very least, have your computer calculator that you can use to wrestle with some problems, okay? Okay, let's get started, folks. So we're in chapter 8, which is one of my favorite chapters. It's about microbial genetics, okay? All right, so in the notes that I will send you later this afternoon, I've already put a video link for a DNA replication video on YouTube. I anticipate that I'll be showing this video to you today during our lecture period together at some time. I'm not going to start the class that way. I just wanted to highlight that there is a video link in today's notes for you to reference. Okay, so microbial genetics. We need to start with some definitions, okay? If we're going to make heads or tails out of this chapter, which is a big, meaty chapter, we need to have some definitions down. Okay, so the first definition is genetics, right? Let's start there, okay? Genetics is the study of heredity. Now, if you don't know what heredity is, then that doesn't mean anything, but let's build upon our conversation here. Okay, so let's define heredity. Okay, heredity refers to the genetic material that is passed from one generation to the next. The genetic material that is passed from one generation to the next. Okay, all right, so let me ask some of you, okay. Okay. In the chat window, can you give one type of molecule that is transmitted between generations? In other words, you know, when your mom and dad procreated and had you, what molecules were in the fertilized egg cell? What types of molecules are we talking that are transmitted to the next generation? Okay, so Kit has one there, absolutely. Okay, so chromosomes, right? Chromosomes would be a very classically correct answer to this question. Chromosomes are genetic material, and of course, you know, Taylor says about DNA. We know that DNA is a component of the structure we call chromosome. So chromosomes, which themselves are made of DNA plus protein, right? Okay, so that would be an example of genetic material. Is there anything else that we should consider as genetic material? Elena, ribosomes, very nice, okay? Now this would not be something that is talked about in a traditional sense. When we talk about genetic material, most people give the very traditional textbook answer of DNA and RNA, because DNA and RNA are nucleic acids, and we know nucleic acids are given to the next generation, right? And to produce your body plan of the person you became, and for your children, their body plan, the person they became, or if it's a bacterial cell, the kind of cell that will be created from the mother cell. Okay, so RNA is certainly relevant. But you know what? In a fertilized egg cell, the cytoplasm of the mother's egg cell is going to have a variety of materials in there that are going to influence the outcome of what the next cell becomes, and if that cell is an animal cell, as it develops into tissues and organs, and ultimately a complete animal or human being. So my point here is that from a very narrow perspective, we could think of DNA and RNA as the only hereditary material, but I think that would be a little nearsighted. From a broader perspective, we can think of any category of molecule or machinery that ends up in the fertilized egg cell's cytoplasm. So DNA, RNA, proteins, a variety of lipid, sugars, the machinery in the fertilized egg cell, which includes ribosomes, as Elena has put down. Okay, so we can put down, you know, machinery like ribosomes. Basically, any content, any molecule, any machine that is present in the fertilized egg cell cytoplasm. Now, some of the molecules and some of the structures that get transmitted to the next generation of cell are transitory in nature. In other words, they don't last very long, but just because they don't last very long doesn't mean they don't have an impact. Okay, some of the RNA molecules that get delivered to the next generation cell, the fertilized egg cell that becomes the child ultimately, some of those RNA molecules, they only last for a matter of a few hours, but they have a deep impact on the pattern of gene expression that unfolds like a stone hitting the surface of a pond and having a rippling effect, a ghosting effect, because that pebble hit the surface of the water, okay? All right, so I'm asking you to be a little bit broad-minded right now in our conversation. Okay, good job, folks. Plasmids absolutely would be genetic material for bacteria, Kit. Absolutely, plasmids, we could put that down there, okay? So we're just talking in broad swipes right now. Okay, let's continue. So genetics is the study of heredity. Heredity is the genetic material that's transmitted from one generation to the next. Traditionally, that's DNA and RNA. But if we're honest with ourselves, the fertilized egg cytoplasm contains a variety of other things that also move to the next generation. All right, well, chromosome. Chromosome, again, is a biological structure made of DNA plus protein, right? We know in animal cells, I'll just say human cells and make it human cells, right? That's going to be DNA plus histone protein. But as we've been learning for bacteria, which are prokaryotic, that's going to be DNA plus non-histone protein. Okay, so human cells are going to have a chromosome that has a traditional X appearance to it. And bacteria is going to have a chromosome that's going to have a folded up or wrapped circular chromosome appearance. All right. Let's talk about genes. Let's talk about genes, okay? Or gene, okay. So folks, let me ask you in the chat window, how would you define a gene? How have you learned the topic of gene in other courses or in other experiences you've had? If you just want to read it out of your book, that's okay, but how would you define a gene? See, the bad thing for me talking about content that you're not getting ready to test on is that you may not be reading that far yet, and I understand. Okay, all right, so thanks everybody for contributing responses here. I love that, and I really appreciate it. Okay, so Chelsea says characteristics of an organism that can be expressed. Okay, I appreciate your participation, Chelsea. I really do. Kit says pieces of DNA that code for various things. So, I know what code means, but does everybody know what code means? Elena, genetic expression. Photomata, heredity passed from parents to offspring. Okay, thanks everybody. I really appreciate that, and feel free to continue to contribute. Okay, thanks, Arlen. Small units of DNA that contribute to what makes up our heredity. Defining a gene is a slippery slope. And what I mean by that is, yes, you can read the definition of a gene in your textbook, but that's only one definition of a gene. Defining a gene is a little bit like defining the word love. If I asked you to define the word love, you would all give me, I don't know, 18, 19, 20 different, but arguably comparable definitions of love. They wouldn't probably be identical, but that doesn't mean they're not all accurate. So how do we talk about a gene here? We need to keep an open mind. I think the best way I can do that is to give you multiple definitions of what genes have traditionally been considered by biologists. Okay, so I'm going to give you the historical or classical definition. Okay. The historically correct definition is that a gene is a sequence of DNA nucleotides that tells the cell how to manufacture a protein. Okay, this is what I was taught eons ago, right? When we just had chisels and stone tablets to write on, right? So a sequence of DNA nucleotides that tells the cell how to manufacture protein. This is an okay definition. It's an okay definition. But, you know, a more modern or contemporary definition is any sequence of nucleic acid, DNA, or RNA that tells the cell how to manufacture something from it. Okay, so, you know, in the chat window, the definition that Kit gave for a gene was something something that codes for something. So when you see that language, okay, when you see the language codes for, you know, what the heck does that mean? It means that the molecule contains information that allows the cell to make something from that information. So codes for means when a molecule contains information that can be used by the machinery of a cell to manufacture something. Okay. So with the more modern definition of a gene, any sequence of nucleic acid, DNA, or RNA that tells the cell how to manufacture something from it, right? I mean, DNA, so my next point is be flexible on your understanding of a gene, okay? If a piece of DNA is used by the cell to manufacture RNA, then that piece of DNA is a gene. Okay. Now, you know, even today, you know, I teach a sophomore level genetics course. We really have to wrestle with the historical definition of a gene, because that's been burned into students' minds for such a long time that, you know, that's what a gene is, a sequence of DNA that tells a cell how to make a protein. Well, it is, but there's more to the story. Okay. If a piece of DNA is used by a cell to make RNA, that piece of DNA is a gene. If a piece of RNA is used to make DNA, that piece of RNA is a gene. If a piece of RNA is used by a cell to make protein, that piece of RNA is a gene. Okay, you see the commonality here is that there you have a molecule which contains information, and that molecule will typically act as sort of a template of information that the machinery of the cell will then decode in the process of manufacturing another second different new molecule. So if you make a piece of RNA, then the original DNA was a gene. If you make a piece of DNA, then the original molecule was a gene. If you make a protein, then the original molecule was a gene, is a gene. Okay. All right. So, you know, if you've had some basic biology, you appreciate that, you know, typically DNA codes for RNA, which codes for protein. That's a very traditional way of thinking about things, and we're going to talk about that here in our course. But we need to be flexible, because in the field of microbiology, some genes are made of RNA and not DNA. Okay, so this second one here that I put into your notes, okay, if a piece of RNA is used to make DNA, that piece of RNA is a gene. Here's the deal. There are many viruses out in the world whose genome, whose genetic material, whose instruction manual is stored as RNA instead of DNA. Now, we think about DNA in our cells. DNA is the genetic repository, but in many viruses, RNA is the genetic repository. So, for example, HIV, the virus that causes AIDS, has its genome made of RNA. So HIV has genes that are made of RNA. Okay. So a typical cell, like the kind of cells we find in our own body, whether it's a human cell or a bacterial cell, yeah, you're going to find DNA, and DNA is going to have lots of genes in it. But to be flexible with how life exists on our entire planet, we can't ignore some of the more important exceptions to that, such as viruses. Okay, folks, are you okay? Any thoughts, any questions? Okay, all right. Well, let's continue here. If you have your digital textbook available, or if you're using the physical book, on page 205 of the physical book, you have this thing called the central dogma. Okay, and we're going to be talking about that. Okay, let me make a bullet point here. Let's see, number 5. Okay, the central dogma. Okay, the central dogma is this basic diagram. Okay, DNA is used as a template to make RNA, which is used as a template to make protein. That's the basic flow of genetic information in a cell, not necessarily in a virus, okay? Okay. This is the basic flow of genetic information inside cells, okay? Exceptions exist, okay? All right, so let me ask a couple questions here. Question 1, or maybe I'll do this, question A, okay? Let me take a look at the chat window here. So Kit asks a question. Since messenger RNA vaccines give us instructions on how to build certain things, so I'm presumably you're talking about the COVID vaccine, are they technically putting genes into our systems? Yes, Kit, they are putting genes into our cells. Please understand, those genes are temporary. They do not enter the nucleus and fuse with our genetic material in any way that we are aware of. But yes, they are putting transient, temporary pieces of RNA that are being used as templates to make viral proteins. So they are putting viral genes in there. Okay. Okay, any other questions? Okay, here's my question to you. I'll put this into your notes, okay? And I just ask you to contribute any responses in the chat window. What is the name of the process that cells use to make RNA from DNA? What's the name of that process? What's it called? I mean, it's something we're going to definitely talk about here in chapter 8. Tiffany, I see your question. I'll answer that in just a moment, okay? What's the name of the process cells use to make RNA from DNA? Anybody remember that from their basic bio lessons? Okay, all right. Hey, thanks, folks. You did good there, right? It is, in fact, transcription. That's right. Transcription is the process of taking DNA, using it as a template to make RNA. Okay, next question. Let me see. Let me answer Tiffany's question in the chat window. Tiffany asks, so the vaccines are not live active viruses, they're just chemically made up similar replicas? That is correct, Tiffany. Well, the topic of vaccine production is a big, complicated one that we could do a whole lecture on. But to answer your question, Tiffany, in the case of the coronavirus vaccine, that is not a live virus they're putting into you. What they're doing is putting some coronavirus RNA, which is coronavirus genes, into a tiny lipid bubble that then enters your cells, goes into the cytoplasm, where your human cell ribosomes translate those viral genes into viral proteins, which then stimulates the immune system to build a defense against the coronavirus. So in the past, and even today, some vaccines have used attenuated viruses, full viruses that have been damaged and weakened. But I think increasingly going forward, you're going to see the vaccines produced using the technology of COVID, which is simply putting in fragments of the pathogen into the cell and stimulating the response that way. Okay. See what kind of good questions come up from this conversation. That's awesome. I love it. Please keep throwing the questions out. Okay. Question B. So we've answered question A. What is the name of the process that cells use to make RNA from DNA's transcription? Here's a question. Can transcription go in reverse? Can a cell take a piece of RNA and reverse transcribe it? Okay, all right. Hey, good job. The answer is yes. Transcription can go in reverse. In fact, when that happens, it is called reverse transcription, okay? So yes, this can happen. It is called reverse transcription. Question C. Question C. What is the name of the process that cells use to make polypeptide from RNA? What is the name of the process that cells use to make polypeptide from RNA? What's that called? Okay. All right. So Kit again is sitting on top of his keyboard, right? So he's right there, and that's fine. So that is translation. Okay. That's okay, Kit. I want you to participate. Okay. Translation. Here's a question. Can translation go in reverse? Can translation go in reverse? Can a cell take a protein and build RNA from it? What do you think? Okay, so Kit is out there. All right, and it would seem that many folks are saying no. Okay. Hey, stick to your guns. Even if you're wrong, stick to your guns, you know. Yeah, the answer is no. If it can happen, we are not aware of it. Science does not believe that cells can do this. It's never been observed. We have no evidence of it. So as far as we can tell, the answer is no. All right, good stuff. And since I'm just throwing questions your way at this time, let me ask this one. Okay. What is the difference between a polypeptide and a protein? What is the difference between a polypeptide and a protein? Right in this chart up here, we had DNA goes to RNA goes to protein. In reality, it's DNA goes to RNA goes to polypeptide goes to protein. So what is the difference between a polypeptide and a protein, or is there no difference? Are these the same thing? Okay, so Kit is asking, isn't a protein a bunch of polypeptides? It sometimes can be, Kit, when there is quaternary structure. It sometimes can be, okay? All right, so folks, I love that we're having this back and forth, and it's okay if maybe you're not always right, okay? That's why we're here. Okay, the difference is this: a polypeptide is a chain of amino acids that has not folded into a proper shape yet. Polypeptide is a chain of amino acids that has not adopted its proper shape yet, whereas a protein, to have biological and physiological activity, is a polypeptide chain that has folded into its correct three-dimensional shape. Okay. Okay, good job, folks. Good conversation here. Okay. All right, so this, this is whetting our appetite for some of the things that we're going to be talking about in this chapter. Do you have any questions you'd like to ask? So Chelsea, you asked, is it how they are wrapped? And I think you're talking about polypeptides and proteins there. If by wrapping you mean how they fold into a shape, I could agree with that. Kit's asking, what happens when proteins degrade? If they come apart, are they technically becoming pieces of loose, non-functional RNA or DNA, or is that something else entirely? Yeah. Well, if the protein degrades, you're going to break down into the individual amino acids. Those amino acids could presumably be resumed, could be recycled to build new and different proteins that the cell needs. So Yasmin, you asked, so is it preparing the protein? Yasmin, are you asking me when you build a polypeptide, you're preparing it to become a protein? Is that what you're asking? The answer is yes. The answer is yes. Okay. So let me sneak in. Sorry for giving you a little bit of whiplash there with the notes. Let me do this. Okay. DNA to RNA to polypeptide to protein. That's really a more complete, accurate definition of the flow of genetic information. Okay, so here's what we're going to do. Okay, if you have your book available, take a look at Foundation Figure 8.2. Foundation Figure 8.2, okay. Unfortunately, I do not have a digital copy of your textbook. I just have the physical copy, so I can't bring this up. So I want to rely on you to have your books open. Foundation figure here says the flow of genetic information. Okay, so we already did this with the genetic, with the central dogma. Okay, but they're talking about something a little bit different in Foundation Figure 8.2. Chelsea, thank you for the page number. Okay. They're talking about expression, recombination, and replication. Okay. So expression. Okay. Expression is when the genetic material is being used as a template to make something else. That's expression. Recombination. Recombination, most students have their introduction and perhaps only coverage of recombination in the context of meiosis, when the homologous chromosomes, when the non-sister chromatids of homologous chromosomes exchange and swap material, and you basically, you basically shuffle a deck of cards to change the the arrangement of the genetic material. You're not expressing information, you're simply reorganizing it. When the genetic material is shuffled or reorganized, okay, this can be in a controlled or chaotic manner. Okay. And finally, we have replication here. Replication is when you copy the genetic material. When a copy of the genetic material is made. Okay. Now, folks, this is a really deep chapter, and in fact, it's deeper than I would love that I would love if I could spend 3 weeks in this chapter. I really would, but I can't. I mean, the curriculum is what it is, so I can only give you a couple lectures in this chapter. So what I've decided to do is take these 3 ideas and tell you which ideas you are responsible for understanding and which that I will not test you on. Okay. And so it's easier if I just do it this way. Okay. Let me do it this way. I'll change the color that I'm using at the moment. Okay. So recombination, you are not responsible for on a test. Okay. You are not responsible for recombination on a test. It's cool stuff. It's fascinating stuff, but we would need 3 weeks to get all of this covered, and we don't have it. Okay. So what I'm saying here is that you are responsible for understanding the ideas of gene expression and the idea of replication, but you are not responsible for recombination. I will not ask questions about recombination on your second lecture exam. Okay. All right. Now, and just a gentle warning. Okay, a gentle warning, and I say this out of the love of a teacher for their students. Okay, every semester I have students telling me that DNA replication is the same thing as gene expression, and it is not. You must learn to put a brick wall between the idea of expression and the idea of replication. They are truly different processes. Okay, gene expression and replication are different beasts. Okay, they are different beasts. Please do not think that they are the same thing, even though chances are at least one of you is going to tell me that on a future exam. Okay, gene expression is about using the information, decoding the information. It's kind of like having your favorite movie on a Blu-ray disc. I mean, we're getting too bad that all my technology analogies don't work anymore because I'm old. But if you have a Blu-ray disc with your favorite movie on and you put it in the player and you watch the movie, so the information on the Blu-ray disc is being decoded by the electronics of your machine, and you watch the movie, the visualization, the audio, that would be gene expression. But if you simply take your favorite movie on the Blu-ray disc and you copy it so that your friend has a copy of your favorite movie, right? You could copy the Blu-ray disc without watching it. Well, then that would be replication. You're simply copying it. You're not using it. You're not watching it. It seems like it's a very easy, simple thing to remember. But when we get into the thick of things, there's going to be all this new vocabulary floating around. And all of a sudden, ideas start to get twisted in the brain. Just be careful. You can do it, but just be careful. Okay. All right. So we're going to begin our conversation with okay. Yasmin has a question. So expression would be the original, we could say. Well, I would not be comfortable with that, Yasmin, because, you know, if you made a copy of the movie and gave it to your best friend could then put it in his or her video disc player and watch it, which would also be expression, all right? So we have to be very careful when we think about what original means.
Okay.
All right. Any other questions? Okay, so to get started down this journey we have together, I'm going to start talking about DNA replication first, simply because that's kind of what your book talks about first, starting again on my physical textbook. It's page 208. I see a figure heading, not a figure heading, a paragraph heading that says DNA replication. Okay, so we're going to look at DNA replication. Some of the relevant figures in your textbook are going to be figure 8.3, figure 8.4, figure 8.5. DNA replication, copying the DNA. How do cells do this? Now, I'm going to give you the perspective from prokaryotic biology, bacterial biology. Why? Because this happens to be a microbiology course. But let me tell you, while there certainly are some significant differences between how an animal cell will copy DNA and a bacterial cell, there are also many significant identical ideas and concepts. And that's what we want to focus on right now to understand what's happening. Okay, so I need to give you some background here. Okay, so points to remember. Okay, so these are things that hopefully you're picking up from your study of DNA and RNA and things like that in chapter 2, or hopefully things that you would remember from having another biology course in the past. But let me remind you of a couple things that will make understanding this topic hopefully easier. The first thing is that you appreciate that DNA is a double-stranded molecule. Okay. DNA is a double-stranded molecule. That should not be a surprise, but you might remember that RNA tends to be a single-stranded molecule. Okay, DNA is a type of nucleic acid. It is made of nucleotides, right? DNA is made of nucleotides, okay? Question coming in the chat window. Here we go. What are the three components of a nucleotide? What are nucleotides made of? What are the three components of a nucleotide, right? I mean, this is something that could show up on Thursday's exam because this is content from chapter 2. Okay. All right, so Kit has answered to me. How about the rest of you? All right. In order to move our conversation along, Tiffany says whatever Kit said. Okay, that's dangerous, right? But okay. All right. All right. Chelsea, thanks for participating. There's some part of your answer that's not right. Okay. And Ireland, there's also part of your answer that's not right. So I guess we need to go over this. Okay. All right. So nucleotides are made of, okay, a sugar, which in the case of DNA is going to be deoxyribose, a phosphate group. Okay, so by that I mean a phosphate atom that's typically surrounded by oxygen atoms, and another big ring structure molecule called a nitrogenous base, or a nitrogen base for short. Let me take a quick look here in your textbook and give you a rough page number of what you should be looking at when you think about these things. Okay. So in your textbook, you have foundation figure 2.16, 2.16. Okay. And Thanks, Elena, for the information on page 205, I think you're talking about. Okay, so on the foundation figure at 216, notice that if you have that up, notice you have a phosphate group which is shaded in like a peach colored coloration. You have a sugar which is sort of purple, and then you have these ring structures which are various colors of pink or turquoise. Adenine, thymine, cytosine, guanine, these are the nitrogen bases. Okay, so let's list those out here. The 4 nitrogen bases are, 4 nitrogen bases of DNA are adenine, thymine, cytosine, and guanine. Okay, that's the name of these beasties. Okay, again, back in chapter 8, figure 8.3, figure 8.3, there's a key, an explanatory panel for that picture, figure 8.3. It shows you adenine, thymine, guanine, and cytosine in sort of a Lego brick kind of cartoon orientation. Okay, these are the 4 nitrogen bases of DNA. Let's see. Okay, question in the chat window. Here it comes. If I had 2 molecules of DNA of the same length, one made of all adenine and thymine, and the other made of all cytosine and guanine, which molecule would be harder to unzip? Which molecule would be harder to unzip? Okay, Chelsea, thanks for answering. Chelsea, please unmute your microphone and say hi to us.
Hello.
Hi, Chelsea. Chelsea, do you have access to your textbook right now?
I do. Can you still hear me okay?
You're a little faint, but yeah. So Chelsea, can you look at figure 8.3, picture B? Do you have that in front of you?
I do.
Figure 8.3, picture
Yes,
B. Okay. So Chelsea, let me ask you, when you look at that, how many rows of dotted lines do you see between T and A?
do.
Okay, good. And how many rows of dotted lines do you see between G and C? 3. Okay, good job, Chelsea. Your audio is cutting out, but you gave me correct answer. So thanks, Chelsea. So the answer is that the harder molecule to unzip is going to be that which is made of GC, guanine and cytosine, because there are 3 hydrogen bonds between guanine and cytosine, and only 2 hydrogen bonds between adenine and thymine. Again, this gets back to the structure of DNA that you're studying right now in chapter 2. All right. Okay, so when you consider the double-stranded nature of DNA, there is a pattern of nucleotide bonding between each side of the double helix called Chargaff's rules. Okay, when you consider the double-stranded nature of DNA, there's a pattern of nucleotide bonding between each side of the double helix called Chargaff's rules. And it reveals that every time, I'll put them down here, every time you have an A on one side of the helix, you're going to have a T on the other side. Every time you have a T on the first side of the helix, you're going to have an A on the other side. Every time you have a C on one side of the helix, you're going to have a G on the other side. This relationship that I'm describing is called Chargaff's rules. Okay, so imagine that you have this double helix right now, okay, that's in your notes, and read each side, right? The left side is one side of the DNA helix, the right side is the other side of the DNA helix. And as we go from left to right, you notice that A and T are always associated with each other, and C and G are always associated with each other, okay? It's important to appreciate that A and T are connected by 2 hydrogen bonds, and C and G are connected by 3 hydrogen bonds. Okay. Any questions so far? Okay, next talking point, and need to reorganize my workspace here. Next talking point is number 5. When DNA is replicated, it uses a process which is described as being semi-conservative. Okay, DNA replication is described as a semi-conservative process. Now, what the heck would we mean by that? So let me ask you a question in the chat window here. Okay. Here we go. If you invest your money semi-conservatively, will you put all the money that you have into the stock market? Okay. So hopefully the obvious answer is no, right? You're going to hold something back. You're going to hold something back because you are not interested in losing everything you own, right? So with DNA replication, here's the basic idea. And for this, I'm going to have to go to the next page and bump down with some note taking here, okay? So if we think about replicating DNA, and we're just getting into this, so I'm not going to go into detail at the moment, but if you talk about a double-stranded molecule of DNA, right? So here's 2 strands, left and right, of the double helix, okay? So here's the ladder, if you will, of the double helix. So we have 1 double-stranded DNA molecule. Then what it means is this. When the cell performs all the chemical gymnastics that it needs to perform in order to copy this 1 double-stranded molecule, you're going to end up with 2 double-stranded molecules of DNA that are going to look like this. Okay, so the arrows are showing what's happening. So we copy this molecule, and as we do so, we're going to end up with this. Where we now have 2 molecules of DNA, where each molecule of DNA has 1 strand from the original DNA molecule and 1 newly manufactured strand, which I'm color coding red. Okay, so the black is the original strand and the red is the newly synthesized strand. Okay. Newly synthesized half. Okay. And the black represents the original piece of DNA. So when cells copy their DNA, they use a process which is semi-conservative because the end product of copying that never results in a molecule of DNA which has both of the original strands of DNA or both of the newly manufactured strands of DNA. That doesn't happen. What happens is you use 1/2 of the original molecule to form each of the 2 new strands, double-stranded DNA molecules, okay? So it's easy to get the words twisted up in there, but hopefully graphically you can see what I'm trying to explain here. Okay. Question in the chat window.
So so just because I want to make sure I understand this correctly. So it's almost as if you took like the very top line part of DNA and then you made the 1/2 and then the bottom part of the original DNA and made the other half. Correct?
Yes, that would be a decent statement. Yes. Okay. All right. Other questions? And so part of our conversation has to be, you know, over today and the next couple lectures, how is this actually happening? Okay. All right. So a couple other talking points here. Let me try to shift back to note taking. Number 6. Okay. DNA as a molecule has directionality to it. A DNA as a molecule has directionality to it. Okay. You might substitute the word for directionality, you might substitute the word polarity. Although I'm not talking about the same kind of polarity I was talking about when we talk about water being a polar molecule. It's a different use of the word polarity. Okay, so let me see here. Okay, what I want you to do is look very carefully at figure 8.3. Look very carefully at figure 8.3. Look at picture B on the right side. Figure 8.3, picture B. Notice we have an example of the chemical structure of DNA. And if we scan our eyes along the left strand of that helix, notice in the top left, I have, and I'll put this into the chat window, in the top left, do you see what I'm talking about? You see the phrase 5' N, 5' N. Do you see that? Do you see that? Okay, thanks, Alicia. Great. Now, on the left-hand side, scan your eyes to the bottom and you'll notice this phrase, 3' N, 3' N. So on the left side, top to bottom, we have 5' end on top, 3' end on the bottom. Now, move your eyes to the right side of the helix and go to the top. On the right side of the helix, at the top, what phrase do we have, 5' or 3'? Okay, good, good. You're seeing it. That's exactly what I want you to appreciate, okay? So if I just stop sharing my screen momentarily, if you look at my hands, if I have 2 strands of DNA, which are represented by my hands, if I did this, that's the directionality, the orientation of the 2 strands of one molecule of DNA in relationship to each other. And the idea is that there is one side that has 5' N, and on the opposite side, it's the 3' N. In other words, they run anti-parallel to each other. Okay, let's get that down in your notes. DNA as a molecule has directionality to it. Okay. So if I do it this way, okay, so if I, rather than top to bottom, if I go left to right, I could do this. I could say 5' on that side and 3' on this side, and then I would have 3' on this side and 5' on the right side. Okay. This is the directionality I'm talking about. when we talk about 5' N and 3' N, what am I talking about? I'm talking about the position of carbon atoms as they are numbered along the sugar. That's what I'm talking about. Okay. In this context, 3' and 5' refer to the numbering of carbon atoms in the sugar molecule. Okay, now we don't need to go any further than that. I mean, I could tell you what that is, but suffice it to say, if you appreciate that the carbon atoms that make up the sugar are assigned numbers 1, 2, 3, 4, 5, and this is something that was developed by biochemists and organic chemists that work on these sorts of things, it gives you a logical way to refer to atoms that are hanging off the carbon atoms of the sugar. Okay, so at this point, I don't care too much if you understand the intimate chemistry, but I really, really do need you to appreciate that the two strands of the double helix run oppositely to each other in an anti-parallel fashion, and that anti-parallel fashion can be explained as having directionality. In your notes, the top strand here that I'm shading is 5' on the left and 3' on the right. The bottom strand is now 3' on the left and 5' on the right. Okay. So, folks, are you feeling okay about this? You want me to repeat anything or try to explain it differently?
Can you just repeat what you said? You were calling it anti-parallel what?
Okay, so let me put another talking point up here in your notes. Okay, so this will be letter A underneath 6. Thank you, kid. I see your comment. The two strands of one molecule of DNA run in opposite directions with respect to each other. In other words, the strands are positioned anti-parallel to each other.
Thank you.
You're welcome. Thanks for the question. Anything else? Okay, so let me take a quick look at my notes here. I think we've covered just about all the basic information I need so that we can actually start having a conversation about things. Okay, so now that you have some background about the chemical nature of DNA, you understand about the nitrogen bases, right? Adenine and thymine, cytosine and guanine. You appreciate the chemical directionality of the molecule. One strand is 5' to 3'. The other strand will run 3' to 5' from the same relative starting position. As you appreciate these things, now we can start actually trying to explain how the process of DNA replication takes place. So at this point, what I'm going to do is move to the video and let that run. That way you'll have something to visualize in your mind's eye as we talk about this. Okay, gang. So hang in there. I'm going to run the video. I will say the video, I like the video. It's pretty good. Graphically speaking, it's pretty sharp. About halfway through the video or something, for some reason, some kind of background music comes into play and it's kind of annoying. Try to ignore the background music and just focus on what the narrator is telling us. Okay, here we go. Let's see what we can do with this.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, there we have it. Sorry about the annoying music. It's a pretty video. I mean, it's nicely done from a visual perspective. It's narrated pretty decently. The background music, I think, is annoying, but hopefully you can learn from it. And it's not, this is not a trivial thing, right? I mean, that was 2.5 minutes of torture there, wasn't it? I mean, this is something that after exam 1, you'll have to start digging into. Okay, so that's the process we're going to start going over today. We're not going to finish it today, but we're going to start going over it. Is there anything you'd like to ask simply from having watched the video one time? Okay, well, remember, folks, at the top of these notes, you do have the video link. I put it in your notes. You'll get that email later today from me, so you'll have that resource to study. Okay, so let's do this. Let's see. All right, so that's understanding the process of DNA replication. To understand the process of DNA replication, I need to introduce you to what I like to use as an analogy as the starting lineup. Think about baseball when you have the starting lineup of guys that are going to be out on the field at the different positions. Who are these people? I need to identify the molecules and enzymes that are involved in DNA replication. Okay. All right, so this is going to be a short list. I'm not going to tell you everything because that would be insane, but I am going to highlight, during your private study time, you want to take a look at table 8.1. That's a good place to look. But let me say a word of caution. Okay. In an effort to conserve, in an effort to conserve space in your textbook, what the authors have done is they've taken important enzymes that are involved in different processes, right? Take a look at the table heading for table 8.1. Important enzymes in DNA replication, expression, and repair. So what I'm saying is, not every enzyme in table 8.1 is relevant for DNA replication. So you want to be careful that you don't confuse things here because of the way table 8.1 has been organized. Okay, not every molecule listed in table 8.1 is used for DNA replication. It's just something you'll have to sort of wrestle with, you know, after exam 1 and when you're back looking at this. Okay, let me give you the starting lineup. Okay, first we have DNA helicase. Okay, in table 8.1, it's simply listed as helicase. Okay, this is the name of the enzyme that unwinds the DNA, that unzips the DNA. The enzyme that unzips the 2 strands of one DNA molecule, right? DNA is double-stranded, so you have to unzip those strands during the copying process. Next, we have some proteins here. Let me take a look at your book. Okay. I'm going to add a protein that's not listed in table 8.1, but it's nonetheless present and used, and it's called a single strand binding protein, or SSB protein for short. Okay, single strand binding proteins are proteins that sit down on the newly unzipped strands of DNA and stabilize their structure. Proteins that attach to the newly unzipped single-stranded regions of DNA and stabilize their structure. Okay, I don't know about you, but you know, every once in a while, well, quite frequently, you know, we'll prepare a meal at the house and we'll have some leftovers. And while we have Tupperware, sometimes for whatever reason, we don't have the right Tupperware or whatever. So we'll have something we need to store. So we'll just bring out some saran wrap, pull it off, tear it off, and pull the saran wrap over a bowl or something like that, right? Well, if your life is anything like mine, 9 times out of 10, I cut off the saran wrap, and what's it do? It crumples on top of itself into this nasty little ball, and then I get pissed off and curse. Okay. So what I'm saying to you is, if you unzip DNA and have 2 strands of DNA that are kind of flopping around like limp noodles, if you don't do something to stabilize them, they will actually fold on top of themselves or even rezip together, rejoin, and block the DNA replication process. The purpose of the single strand binding proteins is to keep the single strands single-stranded so that we don't prevent DNA replication from occurring. Okay. All right. So that's single-stranded binding proteins. Next, we have something that I'm going to call DNA primase. In table 8.1 is simply referred to as primase. This is an RNA polymerase molecule whose job it is to make small snippets of RNA necessary to prime the DNA synthesis reaction. This is an RNA polymerase enzyme that manufactures fragments of RNA needed to prime new DNA synthesis. Okay. All right, folks, I see the clock. I'm out of time, aren't I? Okay. So we'll stop here, pick up maybe next Tuesday. So let me remind you, Thursday, lecture exam 1, log into classmarker.com at 12:30, take your exam. 2 o'clock, please attend lab by coming on to Zoom with me. Next Tuesday, I want to have a discussion with you about some mathematical topics that I've been hinting about in lab to give you some help. So next Tuesday, please have a calculator available. Whatever time is remaining from that conversation next Tuesday, I will continue talking about DNA replication. Folks, do you have any questions? Okay, I have office hours in 15 minutes. I'll be willing to help anybody about any content on the exam that you are concerned with. Pop in and talk to me. Tiffany, please hang on, and I just want to have a 30-second conversation with you. So folks, see you, take care. Good luck on your exam.
Yeah.
Okay?
Thank you.
Okay. Samantha Afalashadi.
I,
Hi.
I'm sorry, did you say the office hour is still in 15 minutes time?
Yeah, 2 o'clock. And if you're coming in to see me, remember there's a different Zoom link that's on your syllabus.
Okay, thank you.
Okay, thank you.
All right. Okay. Yeah, Tiffany. Okay. So have you been able to correct your lack of payment issue with HEC?
So what happened was, so I'm still kind of